Okay. Then the following method is used for the extraction of the juice from the crushed apples. Now there are some errors which you need to notice. So first of all, they take 2 cubic centimeter of enzyme solution to beaker A, and then 2 cubic centimeter of water to beaker B. Now in beaker B there is no enzyme. So what can you say? This can be used as a control. Good. Using a clean stirring rod to mix the content. Why do we clean the rod every time? So that good, to prevent the transfer of impurities or the content of one test tube from to the other. Okay, leave the beaker for 5 minutes. Place a piece of filter paper in each filter funnel. Place one filter funnel in A and the other in B. After 5 minutes, fill the filter paper in A and crush apple enzyme solution from A, and then fill the filter paper in B with crushed apple and water, which is control. Now start the stop clock and measure the volume of any apple juice collected, whether enzymes would gather it or not. Okay. So name an apparatus suitable to measure 2 cubic centimeter other than, okay, we don't use this in chemistry, in bio. Hmm. What else? Hmm. You can take it in something like smaller apparatus. Now think about that, like which are smaller in size and used to collect volume. You can use a syringe or you can use a pipette. Okay. So burette we don't use. Pipette we use, but burette we don't use. Okay. Next one is okay. What should be the unit of time, and check the experiment. Minutes. And what could be the unit of volume? Correct. Because 2 cubic centimeter of enzyme was taken to, so cubic centimeter. Okay, the readings are given. For 10 you have to calculate the reading. Think again. Is 20 till, it's between 20 and 25. Mm-hmm. 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 Wait. This is 21. This is 22. Not 20.2. Hmm. Okay, and the other one? Think again. I can make it bigger for you. Oh, sorry. It's 8.5. Wait, wait, wait. 6, 7, 8, 8.5, 9, and then 10. It's between 8 and 9, so 8.5. Okay? Okay, we have completed the table already. Third one is use the data in table to suggest why the use of this enzyme is an advantage in the fruit juice industry. So we have seen when we have added the enzyme, more volume of juice is collected. Okay. So why do you think the use of enzyme is important or is required? Yeah, okay. Yes. And why do we use it? Why are we using enzyme? Okay. To increase the to increase rate of reaction, what else? Energy efficient, time efficient, and to break down. To break down, okay, less, because since they haven't given us pectinase, so let's make it a general statement. We can say uh, it breaks the content of fruit to release larger volume of juice. Okay. Okay. You can write time efficient, you can write energy efficient. Okay, when we are using enzyme, like we have discussed in pectinase, that since pectinase breaks down the complex sugar into simple sugar, which is sweeter in taste. Okay, and no extra money is required to add sugar. Similarly, no extra effort is required to break the bigger particles since we have added the enzyme. Heat is not required. Simple. Okay. Why do we stir the content of A and B? Correct. To ensure the um yes, yes, they have asked about the advantages. Why the use of this enzyme is an advantage in the fruit juice industry. Okay, for the even distribution of contents. Okay, using a clean stirring rod to prevent the contamination or the transfer yes, of contents. We have already discussed this. Explain how the content of beaker B act as a control. So what we have added in this? So no, it's of 2 mark. No enzyme is added. Okay. So why do we use control? Correct. To have comparable results. So since no enzyme is added, uh, you can say no enzyme is added. Hence, only one variable is changed. Okay. Use the information to draw a table or a graph. Okay, what should be in graph? You have to know about few things. What should be on the x-axis and what should be on the y-axis? Let's write this. On x-axis, we usually take independent variable. Okay, time, and it is dependent variable. Keep in mind the labeling should be related to this table, like time slash minute, and then you have to write total volume of juice. Just keep in mind it's independent variable and dependent variable. Okay. Okay, second is total volume of juice collected per cubic centimeter, right, sorry, slash cubic centimeter, and we have to draw a data for A only. Okay. And even if the points are not like coming in a straight line, still you have to put it into a straight line. Okay, don't make a curve like this or this. It should be straight, as I mentioned, it should be ruled straight line. Keep in mind you have to take your scale with you, pencil, eraser, or sharpener, so that you can make correct points. Okay. Whenever you make points, make a simple dot, encircle it, or I would recommend put a cross. Okay, to have a better result. Okay, use your graph. Okay, since we haven't made a graph yet, but you need to take a printout and make a graph. Okay, take a printout of 1 or 2 pages, which is required, the diagram one, the graph one, and try to complete it. Okay, design an experiment based on the method used by the student to investigate the effect of enzyme on the production of juice. Now take your time, you have 5 to 7 minutes. Read this whole experiment and tell, like, using the same method, how to investigate the effect of enzyme on the production of juice for different varieties of apple. Now the independent variable is different variety of apple. Dependent variable is the volume of juice. Use the same method, think for 5 minutes, and then we'll talk. Okay, so I'm just rephrasing it. So first of all, the different varieties of apples were tested for the enzyme activity. We take the same mass of crushed apple, correct? And then two beakers labeled an A and B would contain the same mass of crushed apple. 2 cubic centimeter of enzyme solution is added to beaker A and beaker B both. A stirring rod is used to mix the content. Uh, then the beakers were left for 5 minutes. Okay, since they are using 5 minutes, we have to mention it. Um, After 5 minutes, the filter paper A and B would be filled with crushed apple enzyme solution from both beakers, and the stopwatch was started, and volume was of apple juice collected in the measuring cylinder was measured after every 2 minutes for 10 minutes. Okay, to have a comparable results. Done. So in addition, you can also write that the enzyme, the volume of enzyme, the concentration of enzyme, and the mass of apple and the temperature would be kept constant during the experiment. Okay, since we have to use the constant variables. Done? Hmm. Now the next question is, a student tested some apple juice for the presence of reducing sugar and observe a red color. What is the name of solution that student has used for reducing sugars? Which solution is used for reducing sugars? Wait, what test do we carry for reducing sugars? Benedict solution. So what would be temperature of Benedict solution, since we need to heat it? You can give a range 50 to 100 degrees Celsius, since we need to heat the content to get the result. Okay, if the color is red, so what is your conclusion? Hmm. Okay, listen. We have carried out the test, the Benedict solution. Yes, we heat the content in a water bath, and the temperature is kept between, let's say, 50 to 100, and the positive result is red, brick red precipitates. It means that reducing sugars are present. So, what is our conclusion? That reducing sugars are present. Okay? Okay. Next one is, okay, a blood sample is given. Identify these cells. What is D and what is C? What is D and what is C? Yes. 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 Yes, the white blood cells and red blood cells. Okay, now you have to make a diagram of C. Can you make it on a paper and send me? Okay. So we discussed already what is C and D. C is the white blood cell and D is the red blood cell and red blood cell. Yes. Done? Okay. Send me now, I'll check. Okay, what you need to keep in mind, the overall size, there should be proper nucleus and the cell shouldn't be broken. Let me check what you have made. Oh my god, it's horrible. Okay, let's say the overall shape of cell is okay, but the nucleus is not okay. Okay, the nucleus is not well in shape. Okay, let's move back to this one. It should be it should be properly curved from here, this point. Okay. And there shouldn't be broken lines or double lines. I've seen the broken lines and double lines. There shouldn't be. Okay. 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 What is the difference between C and D? Wait, wait. There is no nucleus in D, but nucleus is present in C. Okay. Then other difference. D is smaller, C is larger. Next difference that we have done. C contains nucleus, D doesn't. Okay. Okay, how to put this into word? Okay, okay. Look at it closely. Okay, pigment is not there. There are grains. Okay, so you would say that C has grains, while or it has a granular structure, while D is not granular. Okay, so we have to give three differences. One is about, yes, nucleus, no nucleus, larger in size, smaller in size, granular, non-granular. Okay. Okay, next is the length of your drawing, and then you have to calculate the magnification. How to calculate the magnification? What is the formula? Image size over actual size. Let's say image size is the one you're getting from the diagram, and other one is the one which you have made. Okay. Okay, let's move to next paper then.